Hello everyone and welcome to Cellix's webinar, Chemo Pump for Long-Term Cell Culture. My name is Dmitry Kashanin. I'm glad you could join us today. I'm going to start with introduction of Chemo Pump and then we'll show you how to set up Chemo Pump and how to use it and how to use the iChemo application. After that, I'm going to explain you two application examples and we'll finish with quick summary. We are glad to introduce you Chemo Pump. This is a very unique pump which is designed to aid the cell culture of primary cells within the microchannels and flow chambers. This pump is controlled by iPod Touch or iPhone via the iChemo application, and this is compatible with standard CO2 incubator. The controller can be placed outside, and then the pump and the biochip or flow chamber will go inside the CO2 incubator to allow the continuous perfusion of cells with the culture media. Chemo Pump has a number of distinct features and benefits. First of all, it allows the uniform cell growth in the microchannels of flow chambers. It supplies necessary nutrients, oxygen, and CO2. It allows long-term perfusion and long-term cell culture. The media can be pretreated with TNF or some other stimulant, so therefore the cells can be activated. You also can do a pretreatment with high shear stress as the media is continuously perfused through the microchannels. The Kima pump is compatible with standard CO2 incubators, and it will fit on the shelf of those incubators. It's also compatible with V98 series biochips or flow chambers from other manufacturers. The connection of tubing and media to Kima pump is quite easy. To control the Chemo Pump, there is no PC required, and we have a friendly application on iPod Touch. Multiple conditions can be tested at the same time because the pump is supplied with 8-way splitters. Chemo Pump can be used in a number of disease areas: inflammation, cardiovascular atherosclerosis and thrombosis, respiratory and asthma, and also oncology, melanoma cells, and metastasis. There's a number of assays you can execute. You can simply long-term culture primary cells within V98 biochips or other flow chambers. You can look at the shear stress-induced cell culture and proliferation. You can run the toxicology studies where the media perfused through the pump is treated with different compounds. You also can look at long-term effects of nanomaterials on cell growth and cell proliferation. Out of the box, Chemo Pump supplied with the following configuration. First of all, we supply Chemo Pump controller, which is also connected to iPod Touch. Chemo Pump itself supplied with 100 milliliters bottles with special cap, which can connect the necessary tubing. We also supply power supply and the connector, which interconnects the Chemo Pump itself and the controller. One controller can support up to 4 Chemo Pumps, so therefore you can run up to 32 independent microchannels. We also supply Velcro strips so that the controller can be attached directly at the site of your incubator. This is how the controller and the pumps look like. So on the side of a controller you have a power socket. Also on the top you have power switch and LED indicator for the power light and also for the ready signal when the controller is put up and ready for operation. Controller has the universal dock which can take the iPod or iPhone. The Chemo Pump has the Chemo Pump drive, there's also a place to put 100 milliliter bottles where the media can be then subsequently placed, and also there is a connection between the controller and the pump. Each controller can take up to 4 pumps. This is how you actually connect the tubing to the Kima pump. There's 3 tubes. One is a pump inlet tubing, which is connected from the bottle into the body of the pump, and then pump outlet tubing has the 8-way cable attached on one end, which fits to the biochip. Also, there's an option to use the biochip outlet tubing, which is then connected directly back into the bottle. Therefore, this system allows the recirculation of the media from the bottle through the biochips and back into the bottle. There are options actually to use a separate bottle for the waste if it's necessary. The pump is controlled by a simple iKima app, which is installed on iPod Touch. So it has 3 working modes essentially. First page is to connect, and you can connect via Wi-Fi or you can connect via universal dock if the iPod is plugged into the controller. Then on the second page you have a simple washout where you just hit the button and it will start perfusing quite quickly media through the pump and into the petri dish. And the main working mode is perfuse. There you set up 2 times. One is perfusion time and the pause time. So pump can work in start-stop mode or in continuous operation if you set the pause time to 0. Here I'm going to show you how to use Chemo Pump with V8 endothelial plus biochip. And this is a standard protocol we developed for seeding and coating biochips with fusion molecules. So first you coat the biochip with 12 microliters of fibronectin and store that overnight to allow the fibronectin to bind. Then you take the 5 microliter of endothelial cells at 15 million per milliliter concentration and inject it into every channel of the biochip. Then this is incubated for 1.5 to 2 hours in the CO2 incubator. And you can see the image of cells after the incubation. Also, this is comparison of cell spreading after 2 hours in 4 different channels of V8 endothelial plus biochip. Following the initial attachment of cells, the biochip can be set up with Chemo Pump for long-term continuous perfusion. In order to connect the Chemo Pump to the biochip, first the Chemo Pump needs to be washed with 70% ethanol and then after that with water. Then you need to connect the pump inlet tubing and pump outlet tubing to the pump and wash it with media coming from the bottle for 3 minutes. Subsequently, the biochip can be connected and the pump and biochip will be placed in the CO2 incubator. The perfusion can continue from 24 to 72 hours, and this is the image of cells taken at after 72 hours period. As I explained, the pump can be run at in the continuous mode, but also in the start-stop mode. This graph shows the start-stop mode where you set up the pause time and perfusion time. And this can be changed in the dialog boxes. So you can pause, for example, for 20 minutes and perfuse for 10 minutes, or you can have the equal perfusion and pause time. If you set the pause time at 0, then it will continuously perfuse. To date, we have developed several applications for a Kima pump, and one of them is cell culture under flow. Here you can see the image of cells after 1.5 hours incubation and before perfusion, so the cells look not really spread and quite spaced out. And This is the on the right hand side you see the image of cells after 74 hours perfusion with Chemo Pump. So you can see that the cells are much more elongated in the direction of the flow, and also um, there is less amount of gaps between the cells. In another application, we have looked at the metastatic process, and in particular in arrest of the metastatic cells in organs and their migratory behavior and extravasation. And there we have observed the lung cancer cell co-culture under continuous flow with primary endothelial cells. There's an image of 1205LU cell line, which is being perfused through the vein endothelial plus biochip uh, coated with primary HUVEX. And image was taken just after 5 minutes from beginning of perfusion. So there you can see quite a lot of cells attached, but they all rounded up. So we then set up this biochip to be perfused with Kima Pump over 17 hours. There you can see some images of the cells. Now all of them are elongated. Both UVEX have obtained their polarized genotype, and also the lung cancer cells, which are GFP tagged cells. On the right hand side, you can see the image only of the lung cancer cells. You can see that some of them are already polarized, so which means that they are migratory and they are moving around. In summary, Chemo Pump is a unique product. This pump is operated with iPod Touch and has no need for PC to operate. Therefore, pumps can be run in parallel, so customers can use this pump to grow cells in 32 independent channels. Pump employs active flow mechanism, so it guarantees perfusion, unlike some system which can use gravity perfusion. It also physiologically relevant and easy to use. It mimics the in vivo shear flow microenvironment. The pump is easy to use for long-term cell culture on chips, and in conjunction with V9 endothelial plus biochip, it results in experiments which are reproducible and validated. It can be used with a range of different primary cells and be applied in a variety of disease areas. It's also compatible with Vina series biochips and flow chambers from other manufacturers. And you have ability to run multiple channels or multiple flow chambers in parallel. Thank you for your attention.